Professor Kerbel, thank you so much for coming to Milan and opening the Milan 10th anniversary.
My pleasure.
Breast Cancer Meeting. And it was very appropriate, given the talk you gave by Judah Folkman, Memorial to Judah Folkman, to launch the whole symposium, which is stuffed full of Folkman, Kerbel ideas. Now, tell us first about Folkman.
Well, he was a very extraordinary individual because, not just because of his scientific achievements. There are, very large numbers of brilliant scientists around, and he was one. He was also a brilliant and compassionate physician. But I think what really set him apart was his humanitarianism. It was said that when he was a young adult, his father, who was a rabbi, wanted Judah to become a rabbi. And when Judah said no, he actually wanted to go to medical school and become a doctor, his father said, well, okay, become a rabbi-like doctor. And he did. And he also became a rabbi-like scientist. And what I mean by that is having a very gentle, very humanitarian, very mentoring, very open side to him, very, very generous and very, very, very humble. And those characteristics, I think, were instrumental in recruiting trainees to come to his laboratory, not only because of the science and the ideas that he was developing, but they said, well, I want to work for this kind of person. And then eventually some of these individuals began to develop their own labs, and that was a way, of course, of expanding the idea of tumor angiogenesis and anti-angiogenic therapy.
So 10 years ago, really people frowned on, didn't believe, uncertain about the Folkman ideas and the centrality of angiogenesis. And in fact, some of the first papers were rejected by the top journals. That's right?
Yes. I mean, Dr. Folkman used to like to joke about this. Again, he never seemed to be bitter as a result of individuals, granting agencies, other scientists reviewing his papers. He never seemed to get bitter about the fact that he was faced with continual rejection. He would joke about it. He said, well, I know my ideas were not readily accepted because I have all the reprints in my lab from 1971 or from 1979 or 1982. And he used to quote from reviews that he received at the National Institutes of Health when he was first writing grants on angiogenesis and anti-angiogenic therapy, how this was a totally ridiculous idea and it was impossible and it would never come to fruition. But he just persevered. He was convinced that it would be right, and of course, eventually he was proven correct. It took, I would say, probably something like about 14 or 15 years after he articulated his 1971 visionary hypothesis about treating cancer with angiogenesis inhibitor. It took about a decade and a half for things to begin to first turn around with the discovery in his lab of the first pro-angiogenic growth factor. And then a few years later, a growth factor called vascular endothelial cell growth factor, VEGF, was discovered at Genentech by Napoleon Ferrara, and also by Hal Dvorak in Boston. It was called vascular permeability factor. And with those discoveries and the development of drugs to target VEGF, for example, investigators really began to change their mindset. And then there were a series of ups and downs after that. There was a lot of excitement about some preclinical results. Then the clinical trials started, and the first clinical trials were actually uniformly negative. So that created a big downside. But then in 2003, at the American Society for Clinical Oncology, the first positive randomized clinical trial of an anti-angiogenic drug was reported, and that just changed the landscape dramatically.
So the last 5 years, it's really been good.
Oh, it's...
And you've been reading.
Yeah.
What are you going to talk about tomorrow?
Well, I'm going to be talking about... I'm glad that you asked that question, because I'm actually going to be talking about two different things. One of them being a concept called low-dose metronomic chemotherapy, which is something that I've been working on since the year 2000, and again was stimulated by Dr. Folkman as a result of discussions that he and I had back and forth beginning around 1991. And the idea here is giving chemotherapy in a sort of unconventional way at relatively low doses on a very frequent schedule, maybe every day if the patient is taking an oral drug. And it's thought now that this way of giving chemotherapy may work, in part at least, because it actually inhibits angiogenesis. Whereas in contrast, when you give chemotherapy in the usual traditional way, the highest doses that you can, separated by very long break periods to allow patients to recover from the harmful side effects of chemotherapy, that way of giving chemotherapy doesn't seem to have a major effect on angiogenesis. But this idea of giving constant, regular, close beats of chemotherapy at low doses, metronomic chemotherapy, may be safer for patients, it may be quite efficacious, and that's now being evaluated around the world. And in fact, at this meeting on Friday, there will be a major presentation by Marco Colleone, Dr. Marco Colleone, on a very interesting trial in breast cancer, utilizing metronomic chemotherapy in conjunction with one of these new targeted agents like Avastin. Very exciting results.
And that's from you, and we've learned from you. And we're not very popular either.
Well, there is a connection. For example, we published a paper, my lab published a paper in a journal called Cancer Research in 2006. And in that paper, what we reported was, I think, two important things. The first is that seldom, if ever, in cancer research have investigators working with new drugs actually tested those new drugs, usually in mice, using a model of advanced metastatic disease. And, you know, often what happens is that you get very encouraging, if not exciting, results in mice, and then when you try and translate those results into the clinic, either they fail completely or the results are far, far less impressive. And this has led to a lot of skepticism and cynicism about doing research, therapy research in mouse models of cancer. And it's also led to this idea, well, if you're a mouse and you have cancer, we can take good care of you. And we began to think that's not necessarily true. Because typically when you do a clinical trial testing a new therapy, phase 1 clinical trial or phase 2 clinical trial, it's patients who have very bad disease. They have often bulky, advanced, high-volume metastatic disease in a number of different sites. Often they've been previously treated with other therapies and they have failed those therapies. They have what's called refractory disease. And so we want to mimic that situation of actually trying a new therapy, but in a setting of, if not refractory, at least high-volume advanced metastatic disease. So we have been developing models, beginning with breast cancer. That was published in Cancer Research. And then this week, Cancer Research published another paper that we submitted a while ago on malignant melanoma. Again, a model of advanced malignant melanoma where we can even generate brain metastases, which no one has reported before from a primary tumor site. And the second thing that was important in that paper is we then showed that there was a particular regimen of two different chemotherapy drugs. One's called UFT, the other is called cyclophosphamide. They're both oral drugs. And for reasons that we still don't really understand, if they were given to mice in this low-dose, repetitive, metronomic fashion, that it had a rather astounding effect on controlling metastatic disease, advanced metastatic disease for long periods of time. And once I began to talk about that work, and then once it was published, there were some groups, like here in Milan, who decided to do a clinical trial investigating that kind of combination. In fact, what they used was capecitabine, another 5-fluorouracil prodrug, and cyclophosphamide, and then they combined this new anti-angiogenic drug called bevacizumab to that regimen. And this was a trial involving about 46 breast cancer patients. And the results look very, very encouraging, if not exciting. So that's the connection. We did a preclinical study trying to mimic metastatic disease in breast cancer patients. The results that we got looked rather exciting, and so there was more of a rationale to maybe investigate, to test that type of therapeutic regimen in a clinical setting.